Okay, very good morning to all of you. We are now ready to start day 4 of this FDP. So we have our first speaker ready, and I can see participants joining one by one as usual. So I hope by the time we start, more number of participants will come in. I also can see some of my friends whom I invited to attend these lectures over there. So I wish you all a very, very good morning and welcome to this FDP on genome engineering and genome engineering for environment. So let me introduce you to today's speaker, first speaker, Dr. Utpal Das, who is currently an associate scientist in the Department of Neurosciences at the University of California, San Diego, USA. He is my friend for many, many years. In fact, it's over maybe almost 25 years. We met first in Banaras Hindu University when I was roaming around the country exploring possibilities of pursuing my PhD, and he was there as a master's student. So I'm a little bit senior to him, but this friendship has been there for many, many years. Although we meet very infrequently, we are in constant touch. And then he has excelled well as a scientist in the area of neurosciences. Unfortunately, this FDP, we are unable to share his experiences as a scientist in the neurosciences, but he will be speaking on interesting things on genome engineering today. So as I was telling you, he was there in Banaras Hindu University when I met him in one of our common friends' room. So after completing his master's in zoology from BHU, Dr. Das obtained his PhD in biophysics from the All India Institute of Medical Sciences, New Delhi, where he worked on biophysical characterization of protein aggregates in neurodegenerative diseases. Later on, he joined the National Institute of Health, Bethesda, USA, for postdoctoral research to study the structure and functions of neurotransmitter receptors. Subsequently, Dr. Das joined the University of California, San Diego, where he has been focusing on understanding the role of various regulators of neuronal protein trafficking in neurodegenerative diseases, using genome editing, antisense oligonucleotides, and live imaging on human stem cell derived neurons. Currently, Dr. Das is examining altered protein trafficking as one of the triggers of Alzheimer's disease and Down syndrome. His work has been recognized and highlighted in several scientific journals. Dr. Das was awarded NIH Postdoctoral Fellowship and Alzheimer's Association Scholar Award. He teaches neurobiology, stem cell biology, and genome editing to students at various levels. I now welcome Dr. Das to deliver today's lecture, and I hope participants will enjoy your lecture. Let me check out how many of them have joined. Yeah, we have now around 56 participants. There are around roughly 133 participants, but they don't log in at the same point of time. But at a point of time, we have found out around maximum 90 people joining together. Yeah, thank you, Dr. Das. The stage is all yours.
Okay. Okay. So thank you, Professor Bora, my favorite friend Utpal Da. First of all, thank you for actually giving me this opportunity to talk and share some of my experiences. Probably in future, we'll also discuss about the work that I have been doing at UCSD. Also, I think all the participants, very good morning to you. I believe in the last 4 days, all of you, I think you are kind of now very familiar with this genome engineering technology. And I consider all of you are kind of a genome engineer by now. So I promise you, I'm not going to, bombard you, all of you, with so many complicated graphs or calculations or, sophisticated technology today. it will be a relatively, easy to understand, easy to follow. It's a kind of a story. You can stop me anytime if you want, but relatively it will be just a kind of not that scientific. It's more of philosophical discussion that we are going to have. And I believe at the end of my talk, all of you will be thinking what to do next. And I believe many of you or many faculties at IIT, all the IITs or even specifically IIT Guwahati, you are doing genome engineering or planning to do. So I believe some of the topics or notes we carried on with us today will help you, or at least help you decide what to do and what not to do. So as I was given the topic, I'll be discussing mostly on the safety, specificity, and the challenges of genome engineering that currently we face. Okay, so this is kind of a content, or we can say it's a learning outcome, what we'll be discussing. First of all, we'll ask the question, all of us ourselves, the reason for gene manipulation. Really, do we need to manipulate genes? And then little bit or a summary of different elements or components of genome engineering that is actually required to manipulate genes. And as I mentioned, and I believe my previous speakers in the last 4 days, they have, generously explained all different components of different nucleases. So I will not take much time describing them again, but little bit or maybe 1 or 2 slides on that. And then we'll jump onto the safety issues, and we'll discuss what are the key safety concerns we have right now with genome engineering. Then another really critical thing that we're going to discuss, that the off-target effect and some of the potential technical remedies that we can think about. Then also we'll discuss a little bit on not on technical safety, but something on handling genome editing tool that is more a personal thing. And then something on medical monitoring. Then we'll discuss a little bit on specificity. And specificity and safety actually, they're separate words, but they are kind of complementary to each other. If there is more specific technology, there will be less safety concern. But still, I'm trying to segregate these 2 different words, and just for learning purpose. So I'll discuss a little bit on the specificity, especially with the endonucleases, and then a little bit on the target specificity, or how we target a specific organ or a specific plant or a specific cell or genome editing. And then finally, and I think that is most important, the challenges right now we are facing, or the world is going to face after using these genome engineering tools. So we'll discuss key issues related to that, and then some of the affordability or probably availability of genome engineering to across different population in the world. That also will bring in another important issue to discuss, is whether we should stick to only somatic gene editing, or we'll also go ahead with germline gene editing. So these are some of the major points I thought that I should mention to you. These are minor things what we are going to discuss. So first question comes, whether is it really essential to manipulate genes? As we know, genes influence health and diseases, human traits and behavior, and as well as productivity of crops. So researchers over the years, just kind of work on, and is just the beginning to use genome engineering to unravel genomic contributions to all these different phenotypes. And as they do so, scientists are kind of discovering a variety of other potential applications for this genome engineering technology. Of course, there are possibility of human genetic engineering that raises numerous ethical and legal questions. However, as of now, we do not really have a clear answer to all those questions nowadays arising. But you like or you dislike, we are going to manipulate genes. So genes will be manipulated, and it has been done for ages and will be done. But in the last maybe 7-8 years, we are kind of reaching the peak probably. Everybody is doing more or less every other lab, at least in Western countries, and I believe in India also, started doing gene manipulations for different purposes. So obviously, this is really important, and we can say that gene manipulation is critical to cure many human diseases. And when we talk about diseases, largely it comes to our mind some of the most common diseases in society, or we have seen in our friends and near and dear one. And if we talk about gene editing and therapy development, then our brain first it clicks towards, let's say, words like cancer. Different cancers are being targeted with genome engineering, or maybe immunodeficiency syndromes. So all this frequently seen diseases are being treated or being targeted. But today I'm going to argue a little differently in the sense, we know that cancer, it needs treatment. There's a large population in almost all the countries who have cancer patients, and they definitely need better therapy or improved therapy. But there are other classes of diseases also. And today I'm going to tell a little bit about rare diseases before we go into the safety concerns. But this, as you know, this rare disease words, even though it's a rare disease, it's called rare disease, they're not that rare as we think. So rare diseases, it's termed rare because if the frequency of those individuals is less than 200,000, then theoretically, they are called rare diseases. So far, nearly 7,000 rare diseases have been reported all over the world. Imagine the number, 7,000 rare diseases. Hardly we have heard many of them. If you see, let me. So this is just this list of few rare diseases. Let's say, acromegaly. Acromegaly, we have been hearing this name since our school days probably, or in class 2 level textbook. It has some issues with fingers, human fingers. Then we have heard about amyotrophic lateral sclerosis, autism spectrum disorders, Graves' disease, which is a kind of immune-related disease. Then I, for the first time, heard about Madras motor neuron disease. So I never heard about Madras motor neuron disease. So I looked into it a little bit. It's very interesting disease. It has been reported maybe 200 people, concentrated in one area in Chennai. And it's largely a genetic disease, but environmental factors are also associated with this kind of diseases. So what happens in those people, there is a kind of a weakness in nerves, and that leads to very thin and underdeveloped legs. So this is Madras motor neuron disease. And interestingly, as I mentioned, it's only concentrated in Chennai, but there are a couple of cases reported in Italy, maybe in Spain or in China. But interestingly, they have tracked them, and those individuals which have been identified in those countries, they have a descending, or they have a link to Chennai. So another disease is Marfan syndrome. Marfan syndrome, it's basically leads to enlarged or aneurysm in aorta. So it's a dangerous disease that the human aorta, it enlarges and sometimes it ruptures, and that leads to sudden death. And then myasthenia gravis, another autoimmune disease, muscular dystrophy. So list is kind of a never ending. So these are all more or less, we have clubbed it, or FDA or NIH, they have clubbed it is like a rare disease. But even though they're rare, they're not so rare. And India, we are not lagging behind. So far, there are maybe another nearly 300 rare diseases have been reported in India, our country, right? So if you further dig down a little bit into these diseases and see the frequency or the number of people affected with this kind of rare diseases. Look at the numbers in India here. Almost like 72 million people are suffering from one or the another those rare diseases. Let's split furthermore and then look at the statistics state-wise. So look at the states like Andhra and Gujarat and Maharashtra and UP and West Bengal as well. We have like 5 million people, and Assam, we are not lagging behind. Almost 200. We have a great number, 2 million people, basically, kind of suffering from some kind of rare diseases. So why am I talking about rare diseases today? We have 7,000 rare diseases all over the world, probably different kind of rare diseases, and many of those diseases are genetic, and some of them are of course genetic, but you see them once the environmental factor starts working on it. But so far, unfortunately, only less than 5% of those rare diseases have treatment, or at least FDA approved treatment or therapies. And cost is kind of astronomical. So if you see average, I would say $50,000 per year per person it needs as of now to treat one of those rare diseases. So look at the burden society is having. Less than 5% of rare diseases have kind of a treatment, and out of 7,000, almost 6,000 plus, they don't even have any kind of treatment. So if you look at the pipeline of a drug discovery, so you see that just to develop one treatment or one medicine for one particular disease, it could be any disease, maybe a cancer or a rare disease, it takes nearly 50 years of struggle and research. And not just time, it takes more than $1 billion to develop one disease. So that's why when society is having so much a high percentage of rare diseases, and many more probably will be discovered sooner or later. So we definitely need something different from traditional treatment therapy. So that's why it's kind of demanding time so that we do some additional research or clinical trial for these kind of diseases. And interestingly, gene manipulation or genetic engineering has that potential to manipulate, such defective gene. So that's why I'm emphasizing that, some point we like or dislike, we need gene editing tools, and especially people who are having these kind of diseases certainly need gene manipulation. So that's why I thought that, okay, I should introduce something like not just a cancer or HIV or something else, but some kind of diseases, some of them we definitely never heard of it. So now, when you talk about genome manipulation, it is not something new. We are not doing in 2020 or 2017 or 2018. People have been doing genome manipulations for the last several years. So in these two examples I'm showing, they are done in like mid 80s. So when scientists first started inserting DNA sequences into human chromosomal DNA, right, by using homologous recombination technology. Of course, that time it was not known or well developed, but at least they tried to manipulate gene. And then later on, you can see that that frequency of genome manipulation kind of increased, and scientists developed few tools probably and led to better recombination in mammalian genome. And Mario Capacci, who got the Nobel Prize for genome editing, and these are some of the papers they published in 1985-86. So this is not a new technology what we are having now. People have been doing that, but that time it is kind of a very slow process. Can you hear me? So that time, obviously, this was a very, very low frequency event. If, that time scientists were trying to manipulate genes, if they take, let's say, 1,000 cells, there will be only one cells that is kind of gene edited. So obviously it is kind of a not, even though it's kind of opened up the window, but that was not efficient. That was happening in 1980s. So obviously people have been harnessing different tools and technologies, and in 1920s, it's kind of every other day people are doing, every lab is doing high frequency, laser focused gene editing nowadays, right? So how people are doing that? Within the last 10, 12 years, scientists kind of harnessed many players that are actually involved in gene editing technology. And those players, just like this great players, right? They are kind of they're thoroughly tested. They're for their efficiency and their efficacy. And today in 2021, we are able to almost precisely edit genes. So what are those players are right now? The players are our endonucleases. And as I mentioned, and I believe all of you kind of accustomed with all these words, meganuclease, zinc finger nuclease, talents, and CRISPR-Cas9. I think there is a talk today by Dr. Rugul Maut. He will be describing the CRISPR-Cas9 technology. I'm not going to touch that part, but I'm sure all of you have a nice idea about how these different enzymes they work together along with different guides to modify genes, and they are more or less they are really efficient. And if you see in the last 10 years or so, they have been developed such a way that almost like they are at the clinical or preclinical stages now. And technologically, if you see, more or less they have the similar technology. They recognize certain size of genomic DNA, and then they will clip the DNA and then repair it. But if you consider the age of re-engineering genomes, there are certain differences as we go on now. So we have megatels or zinc finger nucleases. That time it was very difficult to use those nucleases, but when talent came, CRISPR-Cas9 came, they're very easy. Trust me, they're very easy to handle, very easy to manipulate, and their on-target activity also it's nowadays kind of refined and refurbished. So on-target activity has significantly increased, and not just that, their specificity. It's still being questioned, but definitely it has improved in the last 10 years. So different nucleases or modified nucleases are being tested every other day in many labs. So we are moving, in the right direction. So if I talk about the basic strategy of genome editing, whatever the nuclease you take, it's kind of more or less the same. So you have the scissors, that means those are the nucleases. And if you want to mutate a point mutation or a single base or let's say few hundred nucleotides, all it will do, some guides or zinc fingers will piggyback. Basically, it is kind of a zip code that where to send the nucleases. So guide RNA will work as piggyback, and it will go and bind to that certain DNA where nucleases will go and create double-stranded break. So once double-stranded breaks are created, what will happen? As you know now, that native mechanism or cellular repairing mechanism will try to heal that double-stranded break. So what it will do, it will try to religate those breaks. It's called non-homologous end joining. So that is one way of doing that. But while doing that, cells are, even though we know they are smart, they are not that smart. They will leave certain faults across the modified region. And due to that error-prone repairing mechanisms, there will be certain nucleotide incorporation that will basically give you certain unwanted or maybe targeted stop codons. That's what we say. So if there is a stop codon, then gene will not be expressed after that. So that's how gene silencing is being done. And gene editing, yes, more or less the same technology, but here we kind of just tweaked cell by inserting a template along with your guide and those nucleases. So what will happen, cell, when they repair those double-stranded breaks, it will take the help of a repair template. And as this repair template is already there inside the nucleus, so what we'll do, it will also, our cellular machinery will also incorporate some targeted gene fragment or DNA fragment into the genome. So this is kind of the same homologous recombination, but nowadays what in 1980s those scientists did, nowadays it's kind of very efficient. very efficient. Within 24 hours, you can actually, if you have everything ready, within 24 hours, you can at least see that it's being incorporated, or some genes are silenced or knocked in or overexpressed. So can you hear me? Can you hear me? I see a message, cannot hear. Okay.
No, you are loud and clear. Maybe it's a personal level problem somewhere. Okay,
Okay.
can you continue?
Okay, great. Thank you. So this is just the gist of the summary of what you have learned in last 4 days. So I do not want to explain more on it. So we know the basic strategy. So using this basic strategy, nowadays, all of us, we are, playing with genes. And right now we have a farm. We can call it a gene edited farm, right? So we have kind of a knocked in certain genes into pig embryo. We have now pigs with leaner pigs with leaner meat. We have gene edited rice that gives more production. And not only that, we kind of developed drought resistant corn. I think that the gene name is Agros1 or something. So basically we have incorporated genes or mutated version that allows us to grow plants or crops where there is no water or little water. And not only that, many cattle suffer from many different diseases, and tuberculosis is one of those diseases. So now that we using this CRISPR-Cas9 technology I think in 2016 or 17, a Chinese group, what they did, they basically mutated NRAMP1 gene probably, and they developed few cows. They're resistant to tuberculosis. And not only that, we are also modifying microbiota. Let's say whatever yogurt we eat or as a probiotic, we have now better version of bacteria mixed with those yogurt. So that gives more resistance or immunity. So we have also, not only that, we are actually playing with a mushroom and other fungi so that we can, have long-lasting fungus or mushroom in our kitchen. So basically, we are playing with all the genes to have a kind of a global gene-edited farm with us. And we haven't stopped it there. Within the last couple of years, we have kind of started playing with human, not just other mammal, but human. And probably you already know some of those examples. So usually what we do using those great players, so we do kind of two different ways, manipulate genes in humans, either ex vivo or in vivo. And for ex vivo, what we do, let's say what I have some diseases, let's say, some liver disease or some lung disease, whatever. So I want to rectify myself. What I'll do, I'll take few cells from my liver, culture them on a dish. And if I want to mutate certain, let's say 2 genes in that cell population, I will design my guides. I'll select the endonuclease and throw them on the cells using different technology. Let's say I'll describe few of them later on, let's say nanoparticle or AAV mediated gene delivery. And what will happen, cells will uptake them, they will correct the genes themselves, and then somebody will re-inject all the corrected cells into my body. So this is what ex vivo gene targeting is being done. And not only that, nowadays we are basically injecting systematically, right? Just to push a needle in your blood, and then gene edited tools, all the players, they will go together, and then it will start correcting the genes you want. So it's kind of in vivo, not kind of, it's an in vivo correction. And not only that, we actually not just targeting the overall, all the organs, but we have started, targeting specific organs or specific cell type probably in future. So it's kind of, we are kind of jumping into all different diseases, all different animals and plants. Everywhere we see some benefits. And this list, it's kind of a never ending list. You don't need to worry about this list, but successful, this is just to summarize that wherever, we are trying, we are kind of successfully edited many genes, not only in vertebrates, also in different invertebrates, plants, different approaches we have used, gene knockout, gene knock-in, all different technologies. So basically, our gene gen is out of the bottle. it's kind of flying around and asking for more jobs, because one after another he is correcting the genes and he needs more and more genes, or more and more work, you can say. So basically what we are trying to do, this is just the beginning to modify the most fundamental elements of what makes us human, right? We are going to play with genetic material. We are playing with chromosome. So Jenny is out there, and if he needs work, but this is the time, if we do not control Jenny, he may lead to mishaps, right? And mishaps are not uncommon in gene editing. Mishaps are happening actually. This is just a tiny example I'm giving you. This is a paper published in Cell, I think in 2017 or 18, from Data Eagles lab at Columbia University, New York. So now what they did actually, they were trying to do, they basically wanted to correct blindness. Okay, so to do that, they took a sperm, human sperm. They did this experiment in embryos. Okay, so they took a sperm that has a mutation in the gene called EYS, eyes, and that mutation actually lead to retinal pigmentosa, or basically it leads to blindness. And they took an ovum or an egg from a normal, healthy individual, female, and they tried to fertilize that. And not just they are inserting the only sperm, but they are adding a cocktail of genetic engineering tools. They have the guides that basically targets the EYS mutant gene, and they also injected whatever the nucleus they wanted to use. But the goal of this paper or this work was to correct the mutant, paternal mutant EYS gene at the embryo level, at the 1 cell level, or even at 2 cell level. So that is their goal. And how the CRISPR will work, that once they go inside the cell, in the egg, sperm will fertilize it. It will transfer the mutant DNA, which is a bad DNA. But this genetic engineering tool will use the maternal, which is a good and wild type EYS gene, as a template, and it will correct the sperm's mutated bad gene. That was their goal. Okay, and they successfully did it. Almost 50% of those embryos, they successfully kind of corrected those mutations. However, they also found that chromosome 6 actually, that contains this EYS gene, it's broken. It's completely broken in many of those embryos. And not just broken, in some of those embryos, this entire chromosome 6 was kind of missing. Imagine, this is in 2018. It's not that long ago, just 2 years ago, they published this thing. So what I'm emphasizing, that the mishaps are happening. Even though we have those great players, mishaps are not uncommon. This is just one tiny example. And these kind of examples actually bring in the safety issue for genome engineering, right? So this is the time why, that's the reason why actually we need to discuss safety issues. Now you can ask me a question, right? Why actually do we need to, concern at all when people are publishing gene-edited papers or, CRISPR-related papers at an exponential rate? So I found this statistics very interesting. Many of you probably already seen this. So look at how rapidly evolving this genome editing field is. Look at the papers just with the word CRISPRs in last, from 2002 to 2020, let's say 18 years. In the middle of that, almost 1 or 2 papers per year. But look at the number of papers people are publishing now. It's kind of a true exponential rate. So everyone is trying to manipulate genes. New technologies, new discoveries are coming out, but still we have mishaps like this actually. So it's really the time, not just the, look at the good thing with genome engineering, but also we need to focus on safety issues. So why it is important? Because this entire gene editing technology, it's in kind of an unexplored territory. We are jumping into a pond, we do not know what animal is there. So it's kind of a completely unexplored. And when you jump on the unexplored territory, consequences could be devastating. So unexpected consequences are very common in gene editing. And as I mentioned, that list I showed you, it's hundreds of clinical trials are going on all around the world. I have a slide at the end. Nearly 3,000 clinical trials are being conducted as of now, as we speak today. So expectations are really high from gene editing. We kind of really need therapies as soon as possible. So this is actually it's actually harming us, because as expectations are more, we are kind of manipulating genes without seeing the side effect. And remember, when we use genetic, when we modify genetic code, it is not something that we correct it or mutate with some kind of chemical or kill a pathogen or something like that. We are modifying it permanently. So that's why safety concerns are much more, essential nowadays. And there is no trial and error. There is a trial and error, window which will not be acceptable after a certain point. And safety is the most important thing. Then only we can, bring those technologies or treatment to public and say, okay, I have developed this new genome editing technology. Why don't you use this? And your, friend or a family member can be cured who is suffering from kind of a rare diseases. So safety is really important. So today I am going to touch few of those points. Firstly, technical safety I will describe a little bit, and mostly we'll be discussing off-target effect of genome editing, and then little bit not technical, but that is unintended biological impact of genome editing. Third thing, we'll be discussing a little bit on the handling system, how one should handle or what should be done when handling this kind of tools. And then finally, we'll be discussing a little bit on medical monitoring. Okay. As I said, that almost all the technologies, whatever the endonuclease you use or you have learned or you have heard in last 4 days, more or less they have the same mechanism. Create a double-stranded break at the genomic level and ask the cell to repair it. So most of those are targeted in the sense wherever we send our guide, our nucleus will go and clip there, not some other places. But double-stranded breaks are always taking place in unintended site, let's say here and here on this slide. So those double-stranded, unwanted double-stranded breaks actually are the major safety concern nowadays. So when I'm saying that we are basically creating double-stranded break, more or less, we have the control on it. If not absolute, but at least we have a control. But when cell repairs it, that particular step, we have no control in it. Okay, once you inject, go home, come back tomorrow, cell will do what it needs to do. But we do not have any control over it. So those double-stranded breaks are not always safe. Many of chemotherapy drugs, actually it is doing the same double-stranded break, or it's creating same double-stranded breaks in cancer cells, right? So we do chemotherapy, and those medicines or small molecules go and create double-stranded break, and that's how cells die. But it's very common to see non-specific or chemotherapy drugs attacking non-malignant tissue. So it's not always safe to have double-stranded breaks. Now, this is just a tiny example. You can see that how double-stranded break can actually cause tumorigenic. It could be tumorigenic. So let's say if you have a leaky mitochondria, it could release cytochrome, right? And that cytochrome can activate apoptosis or caspase pathways. And those caspases actually can activate certain kind of DNA. And those DNAs can enter the cell and create double-stranded break. And once those double-stranded breaks are created, certain kinases are activated or certain phosphatases are deactivated, and finally leads to NF-kappa B activation. And all of you know that NF-kappa B activation could lead to aggressive tumorigenesis or induction of cancer stem cells. So basically, double-stranded break and genetic instability and cancer, they are closely associated. So we cannot rule out any further issues if there is double-stranded or unintended double-stranded break happens in the chromosome. So 3 examples I'm going to give you today. Okay, so that could give you some idea of what is happening in this genome engineering world nowadays. And one of those examples is a paper that was published in 2015 and 16 from a Chinese group. So what they wanted to do, very cute little experiment they wanted to do. So they wanted to inhibit or knock down myostatin genes from rabbits and goats. So why they wanted to knock down myostatins using CRISPR-Cas9 system? Because myostatin is a negative regulator of skeletal muscle development. That means if there is more myostatin, muscle growth will be hampered, muscle growth will be suppressed. So if their hypothesis was that you knock down myostatin, then you will have rabbit or goat with more muscle, which is obviously beneficial for economic standpoint. So what they did, they took embryos from rabbit and a goat and injected Cas9 mRNA, that enzyme you need, at different concentration, along with different guide RNAs, sometimes single, sometimes multiple. And then once they're electroporated, they transferred those embryos and basically they allowed for term. And then they have fetus. Of course, some of them are stillborn, have some defects, but at least they have live fetus. Okay, so what they have these cute little rabbits. So on the left here, you can see this is a control rabbit, and that poor guys, you can see the ribs, they're not healthy. Of course, there is a control rabbit. That's what it was supposed to be, right? Nature has developed like that. But look at the treated rabbit, where they inserted guides that is kind of knocking down myostatin along with the CRISPR-Cas9, all the tools. And look at this, healthy rabbit body, right? Look at his muscles. He's kind of almost ready to go to gym and have six packs, right? Look at the bicep here. It's definitely larger than the controlled one. And this is the quantification here. The bicep muscle has definitely significant increase. And not only that, look at the body weight. It has increased in the treated, and further statistics shows that only, they obtained increased mass when they have completely knocked out myostatin gene from those rabbits. So this is kind of an intended and safe application of genome engineering, right? No problem with them. Let them play with those rabbits or the goats. However, when they carefully looked into it, this is what they have seen. So the twin brothers and sisters of this treated rabbit, look at their tongues. Look at their tongues. Many of those siblings, they have large or huge tongue actually. And they happen to be something like this when they grow up. They have huge tongues. They're kind of protruding out. Now imagine what will happen if someone knocks down my myostatin and I gain muscle, I have my new six packs, but I have an enlarged tongue protruding out. That will be definitely, inconvenient. I will not be able to swallow. I will not be able to chew. So basically, this poor rabbits, they have some side effect which is not related to their targeted genome editing. So this kind of genome editing is unintended and unsafe. So one example, the same paper, same group, same rabbit, they have intended and targeted genome editing, but the same experiment also led to development of unintended or unsafe treatment. So this is an issue. That's why we need to discuss safety concerns or off-target effect, right? Second example. So this is a great group. I think they are at the Francis Crick Institute in UK, Cambridge, and along with it's a big group all over the world. What they did in 2017, they published a paper in Nature. So their goal was to look at the embryogenesis, right? We all know embryogenesis. So they wanted to study the role of one protein called OCT4, how this OCT4 actually decides the cell fate. Let's say we have an embryo, only 2 cells or 3 cells or 4 cells. Those cells will divide to form morula or blastocysts, and then each of them will, obtain different fate to generate lung or brain or blood or whatever. So they wanted to, this group, they wanted to know the role of OCT4 in that particular fit decision making. So how to do it? For any phenotypic assay you want to test, what we do, we basically knock down a particular gene or overexpress a particular gene. This is the most common thing every other lab does. So they knocked out OCT4 using CRISPR-Cas9 technology. Very easy. They took out pluripotent stem cells, or they also tested human embryonic stem cells and deployed human zygote, and also they tested in mouse zygote. So what they did, they took the zygotes and injected all these different tools and guides targeting OCT4 gene. And they successfully did it. So what they did, they completely knocked out OCT4 from those embryos. And they, of course, as expected, in those blastocysts, their development was kind of a compromised. Where there is no OCT4, there is a defect in blastocyst formation, which is kind of expected. Then what they did further transcriptomic analysis to look at different genes, what have been overexpressed or underexpressed following OCT4 knockdown. And they have a list of such kind of a gene. We are not bothered about what they observed. But what they mentioned, that this kind of specific or safe knockdown of genes could be employed. And they calculated, they did whatever possible in 2017 to look at different off-target effect. And they conclude that CRISPR-Cas9 mediated genome editing is a powerful method for investigating gene function in the context of human development. Fantastic work, right? Now, look at another paper from the same group after 3 years. It was published in PNAS last year, June. Same group kind of revisited their previous finding that was published in Nature. And this time, they did the same technology, no problem, same kind of injection, but all they used different kind of single cell genomics or transcriptomics methods to look at off-target effect. Previously, probably at that time, they were not available. So as they go by, new technologies are being developed, and they are kind of revisiting their previous findings. And what they observed, that after single cell genomics and transcriptomics dataset analysis, they found that not just OCT4 was mutated in those embryos. Actually, all this CRISPR-Cas9 or guide RNAs were also mutating some other genes, but near to that OCT4 genes. It's kind of unwanted thing, and actually that led to loss of heterozygosity in edited cells, which is kind of dangerous, right? And unintended, look at this. Basically, they also saw a segmental loss and gain of chromosome 6. It's a major problem. So unintended genome editing outcomes were present in almost 16% of human embryos. It's a large population of embryos. So what I mean to say, it is not just the enzyme or not just the guide, but also the detection technology that is critical. In our lab, we routinely edit genes. So we have human fibroblast from Alzheimer's patient or Down syndrome patient, and we try to modify them into, let's say, I have a wild type human neuron. I want to make a Alzheimer's patient neuron. So we do genome editing like TALENs and CRISPR-Cas9 to mutate them. And many times what we observe that, our targeted mutant is there, but when we send our cells for karyotyping, just chromosome aberration analysis, we see kind of chromosomal duplication or deletion. So those kind of things are still happening. It's not that rare. So yes, we need to concern about safety issues. The example 3, I do not want to repeat that. Already I have explained that. This kind of mishaps are there. But if you look at all these 3 examples, 1, 2, and 3, what we see that off-target effect is a major issue here. And accuracy and precision. Sometimes what happens, our guide RNA is carrying our nucleases to the right point, no off-target effect. But what happens, due to certain regions, certain unknown regions, the precision or on-target gene editing is kind of not up to the mark. It's not giving us the desired genetic outcome. So all these different things we need to discuss. And off-target effect, as I mentioned, it can happen due to unintended double-stranded break. And usually, if you see there, those unintended breaks are actually they take place near the on-target site. So if our enzyme and guides are designed to target this region, mostly, if not all the time, unwanted or unintended cleavage are taking place somewhere upstream or a little bit downstream of the genome. But it is just a general thing. It's not always correct, right? And then also off-target effect can happen when cellular machinery, as I mentioned, we do not have any control how cells repair this kind of double-stranded break. And cells can make mistakes. So what happens, sometimes funny things can happen. Let's say we created a double-stranded break, just like any other technology. And then next day you go to lab and you see cell has corrected it, but in a different, it can mix match. What can happen, cell can use two different approaches to cure or repair this kind of double-stranded break. One is, let's say, one strand is corrected by just non-homologous end joining, but another strand can be corrected using a homologous recombination. So instead of just homogeneous population, you have a mix match kind of a repair system, obviously, which can be devastating in terms of cell fate or survivability. Then sometimes what happened, as I mentioned in our own case, first-hand experience, that we do see large deletion or rearrangement of chromosome when we do this kind of genome editing, either TALEN or CRISPRs, doesn't matter what it is, but we do see this kind of deletion and complex genomic rearrangement. This is just the paper, they see this kind of large deletion and complex rearrangement, and that usually happens when your target cells are like embryonic cells or hematopoietic stem cells or even human immortalized epithelial cells. These are kind of low frequency event. It's not that frequent, but it is significant. It can create problems to your experiment or future application of all your gene edited cells. So these are the different reasons why we see an off-target effect. Now you can ask me, okay, Utpal, you said different problems. Is there any way to correct them? Thing is, as I mentioned, this is kind of a rapidly evolving field. As of now, there is no particular way to correct off-target effect. All we can do, use different approach to design our experiment and then select the best one possible. And then, of course, we need to screen them after you modify the gene. So when let's say tomorrow you decide after this FDP that, okay, let's mutate this gene in our lab. So what will you do? You'll design some guides, right? So designing all those guides, nowadays it's very easy. All different institute, let's say from Feng Zheng's lab at MIT, they have this CRISPR.mit.edu, another website at DKFG in German Cancer Center, and different softwares are available. They're really good softwares. They use fantastic algorithm to design, or they will do their best to give you the best guide that will probably have limited or no side effect or no off-target effect. But if you again go back to that cute rabbit with the huge tongue paper, they also use the same technology. They designed their guides at gRNA using this MIT-based web page, right? So it's not always correct. There could be other issues. So best thing, okay, tomorrow if you really decide onto it, use all different websites and algorithm or whatever coding thing you have to mismatch, mix and match what all the guides your softwares are giving and select the best top 5 or something like that. So there is no correct or precise answer for it. And there are new papers. Actually, I found this paper available very recently in 2021. Nowadays, people are actually using deep learning technology. And being at IIT, probably many of the engineers are working on it. So you should collaborate with them and then tell them that, okay, I want this kind of designing system. Why don't you help me? They code, and then this kind of deep learning experiments, I'm sure many of IITs or other institutes are doing. So it's kind of an interdisciplinary approach that we really need to use now to manipulate gene. It's not just a job for a biologist alone. So people are kind of collaborating. So these kind of papers are helpful how to design. And next thing, as I mentioned, double-stranded breaks are actually bad. Is there any alternative to it? Can we actually use or modify a gene without creating a double-stranded break? Actually, we can. So this new Cas9 has been developed. Is basically they do not create any double-stranded break. So what it will do, it's kind of a deactivated Cas9, but it will still bind to the DNA where it's carried out, where it is taken by or guided by HRNA. But what will happen, instead of just cleaving or creating a double-stranded break, what people have done, they piggybacked an effector molecule along with this big Cas9 enzyme. So as guide will say, okay, my dear Cas9, you go and modify the chain. So it will go and bind. But this effector molecule, which is now bound on its back, it will basically either activate the gene expression or inactivate the gene expression by regulating the RNA polymerase access to that particular gene. This is something totally new where you do not really need any double-stranded break. So these are kind of a hope that, okay, future we have a better technology to play with all this different system. Next, if we see here, let's say talk about unintended biological impact. So many times what happens, you try to modify a gene, but instead of, you can modify a gene at certain cells, but not all the cells. In this paper, they tried to modify p53 mutant. So, it is really important for cancer progression. So they wanted to correct this p53 gene using CRISPR-Cas9 system, and they could able to do it. They were corrected, but certain cells, let's say, pluripotent stem cells, those cells were actually not allowing to, edit p53. So cells develop their own way of, nullify the effect of gene editing. And probably once you insert all these different tools into the cells kind of activate certain pathways, which is kind of unintended biological pathways kind of activated, and they will inhibit this kind of modification. So cells are not that full too, right? So these are kind of technical issues I discussed. And then, of course, unintended biological impact, we can't, play with that, because most of the time, whenever we target a gene, we target some transcription factors. So whenever you design your experiment, when you want to, target transcription factors, be aware that you do not play with them, or their inhibition don't, deactivate or activate certain cell survival pathways. So designing is really, really important. Then this is little non-scientific, non-technical issue where safety comes. So let's say tomorrow you have all the different tools, you purchase all the kits and this. Who are going to handle them? Scientists, lab staff, and once they are developed, you are going to hand over to a clinician along with a nurse. Okay, you say that, okay, you have these genetic tools, you inject to a patient X, right? So they are going to do it. But remember, they are going to, or you are going to handle different nucleases, different guides, different viral delivery vectors, reprogram cells. They are not always safe. If you have all the combination together on a tube with a viral vectors, everything, if that spill on your skin, there is a remote possibility that they will be uptaken, because mammalian cells are, they do that thing. There are examples. So this kind of handling issues one has to consider, okay? And then medical monitoring. Tomorrow, let's say you inject certain gene editing tools to one patient to correct certain genes. Let's say, for example, sickle cell anemia, or let's say severe combined immunodeficiency syndrome, right? So if that patient is having sickle cell anemia, you correct the gene, and then if in future certain issues come, then that patient will go to a hematology department who deals with blood or blood-related disorders, or the patient with severe combined immunodeficiency will go to immunology department. That's fine, totally fine. But if the adverse effect or consequences arises due to general or specific gene editing, where will you go? So there must be a kind of a protocol for long-term follow-up when you basically, deal with genome editing tools. So this is, I think, important for us to discuss. And I think we are at a point we really need to concern about different safety issues, right? So I think, okay, we should go to the specificity issue. Specificity, it's with safety and specificity, they move hand on hand. If there is more specificity, there should be less safety issues and vice versa. But still, I'll try to, describe a little bit on specificities, especially related to nucleases. So basically, there are certain issues or certain things we can do with our nucleases to increase the specificity. And also we can develop, or people are developing, different delivery system that will lead to better specificity. And of course, little bit on the duration and the time genome is exposed to an endonuclease is also critical for specificity. But before that, just trivial thing. So whole genome, right now you can ask, is it really, essential to really worried about double-stranded break when every cell is kind of itself is mutating all the time, creating double-stranded breaks, right? So whole genome sequencing is now kind of revealed that humans are born with tremendous genetic variability. Millions and millions of single nucleotide polymorphism per individual have been identified, right? Nearly 500,000 insertion and deletions are taking place in everyone's body all the time. Many create frame shift mutations, and of course, some of them will lead to cancer. But if you look at this single cell level, every stem cell is kind of average 3 mutations per cell division is taking place. And they're doing repairing like 20 to 40 spontaneous double-stranded break. So it's a big astronomical number. Among all those numbers, this kind of 1 or 2 double-stranded breaks created by endonucleases, are they really important? It's kind of still a question to debate, but at least we should be aware about that. So how to increase the specificity of the endonucleases? So I think previous speakers have described that how to, increase specificity. Just, if we just look at the mechanism. So let's say the zinc finger, nucleases. So what they do, those nucleases are basically targeted to a genomic site by zinc finger motifs, right? Those modules, small modules. So if we increase the module length, that will obviously increase the specificity or recognition site. So that's how, zinc finger or talents can be, can be further modulated with better specificity to attack a specific DNA sequence. More the number of modules, better will be the specificity or recognition by this kind of nucleases. For CRISPR-Cas9, instead of just regular Cas9 using, nowadays there are many different Cas9s or smaller Cas9s are available in the market. It's called, SP stands for specificity. So those kind of Cas9s are available that has been shown to have little off-target effect or double-stranded breaks. So these are different techniques one should know when you design the experiment. Now, what about the delivery system? A delivery system could be 2 different things. Once, if you take out the cells from a patient and you want to correct a gene, you culture them on a dish, right? And once they have the cells, what will you do? You just throw your genetic editing tools along with some viruses or liposome or nanoparticle on top of that. And they will go inside, they will correct the gene, cell will repair it, and then you transplant those cells into that particular organ or that individual or that on crop. Similarly, if you have in vivo, then of course you have to have different specific delivery vehicles. Most of them, or most of us, we are actually using either viruses or nanoparticles. Each of those technologies, they have different plus and minus, but overall, if we are talking about delivery system, FDA and NIH, they have basically recommended adeno-associated virus, or in short AAVs. They are actually less harmful or more specific for a target or organ, at least in human or other mammals. So currently AAVs are being used for most of the gene delivery system. However, remember, these AAVs are not always good, because if you want to correct a gene that is involved in muscular dystrophy and you send all your tools with AAVs. They are good. These AAVs, they remain inside the cell for long term. So for a disease like muscular dystrophy, that's a good thing. But those, as you know, they are actually long-lasting enzymes inside the cell. There is a probability they will also start non-specific cleavage. So we have to really think what actually we need and what is our experimental design. And this is just a slide to summarize what all different AAVs or serotypes of AAVs are being used to target different organs, starting from brain to salivary gland in mouse, and in higher order mammals, like different organs are being targeted using different serotypes. In primates also you have specific AAVs, and in human, let's say AAV2 is really popular. AAV9 also is popular. You can target the brain and other different organs. So we are at a stage where kind of have better specificity in terms of delivery and endonucleases. So finally, I'm going to tell you a little bit about the challenges, which is actually really important, equally important for to discuss about in today's scenario. And it's just kind of movie kind of a thing. You have seen this movie, right? The good, the bad, and ugly. So playing with genes could be something similar. Sometimes it's really good, sometimes bad, and sometimes it could turn into be really ugly, right? So we have to be prepared, and we have to get things ready how to deal with these ugly things here, right? So a couple of good things what we can discuss that, okay, using genetic tools or genome editing tools, people are actually curing diseases like sickle cell anemia or sickle cell disease. So sickle cell disease, as all of you know, you have been studying this disease since your +2 level, that there is a change in hemoglobin genes, and that mutated hemoglobin actually aggregates, and that's how it cannot carry oxygen, and cell become sickle shaped. Now, using genome editing technology, scientists have kind of modified this particular point mutation, and now you have a healthy hemoglobin in an individual. And those, ex vivo gene manipulated corrected human blood cells are reinjected into patient, and that kind of now curing the disease. Or sometimes you just activate another, genes. Let's say instead of beta globin, you activate gamma globin using the CRISPR technology. That will also kind of bring in normal hemoglobin. So basically, these kind of good things are happening. Muscular dystrophy, it's kind of almost treatable now. It is one of the first human disease kind of allowed to treat for using gene editing tools. So what happens in muscular dystrophy? There is a frame shift mutation at the exon 51 that leads to a defunct or dysfunctional dystrophin genes. So this non-functional dystrophin leads to muscle contraction, and that muscle contraction actually leads to tons and tons of calcium release, and that calcium actually is toxic to the cells. So cells die. So muscles become weaker and weaker. So in vivo gene editing technology, they have removed this bad exons from this particular gene, and they have kind of almost corrected muscular dystrophy, and healthy lifestyle can be maintained. In our lab, what we are doing, we are using gene editing technology, not on the patient or not trying to correct Alzheimer's, but at least we are at the beginning of how to handle different proteins associated with this particular disease. As you know, Alzheimer's disease is kind of a neurodegenerative disease. So what happened in Alzheimer's brain, you kind of have a dysfunctional or neuronal loss in the hippocampus area. And hippocampus, as you know, this is important for memory storage. It's kind of completely destroyed in elder people. So this is a disease that prevalent in people after like age of 70 or 65. But it's very frequent nowadays. And in India or in Assam, it's many people are actually, I know personally, who are having this disease. So what happens this disease, that there is a protein called amyloid precursor protein. This protein is present in everyone's brain, including yours. Okay, so this protein is cleaved by a couple of secretases, one called beta secretase. And if this protein is cleaved by beta secretase, it kind of generates a toxic protein called amyloid beta protein, which kind of aggregates inside the brain. And this kind of aggregation inside your hippocampus could lead to irreversible neuron loss, or in other words, memory loss. So, what we are trying to do in our lab, we are trying to gene edit the APP gene so that base or beta secretase cannot process this particular gene. And in theory, there should not be any amyloid beta in our brain. So we took mouse and injected our guides and CRISPRs using AAV, as just I mentioned, and tried to kind of manipulate APP gene. And we have some interesting result. We 2 years back, we published this, and this work is ongoing. And overall, if you see, nearly 3,700 clinical trials are going on all over the world. So this is really a good thing, right? There is hope. And not only that, crop production, right? Look at the genetically modified crops across the world, the percentage share, US and Brazil. But look at India, we are not lagging behind. We are doing better. So these technologies are helping us treating different diseases, or helping us to produce more crops. But the bad thing in that movie, Good, Bad, and Ugly, the bad thing is what's the cost or price tag for genome engineering? When I started my talk, I was kind of arguing that we need genome engineering to cure rare diseases, right? But actually, so far, that scenario is not there. Right now, whatever the available therapy is there, they are very, very expensive. That is a bad thing, right? So look at these different drugs and the population they are treating, and the cost of those drugs per year, per person, per individual, almost reaching out to 1 million. So Novartis recently, they kind of developed this drug against the spinal muscular atrophy. This is just kind of a single guide and CRISPR they are injecting one time, and it cures, no problem. It cures the disease, but it costs $2.1 million. So this is the world's most expensive drug, right? So you have the medicine, but what we targeted or what we aimed for, so far we haven't reached there. This is a bad thing. So basically, it is not affordable to everyone. So we can say that, okay, you guys are just targeting a certain population over the world. Is it true or not? So we need to consider that. And tomorrow, let's say Human Genome Project is completely done, right? So, these technologies are very available. Everybody can do their own DNA sequencing in like 1 week time. 23andme.com, they're kind of sequencing genome in like $50. So imagine a situation tomorrow, all of us, we are having our own DNA sequences, and we find certain genetic predisposition in my own genome, or let's say your genome. And that predisposition can lead to certain diseases. If I know what disease I can have, and if health insurance companies, they know that this person X or Mr. Utpal is going to have this disease, here, they are not going to give me health insurance, right? So these are kind of untold stories. They are coming into play. So these are the bad thing. But hope is that, okay, FDA is still working or evaluating almost like 1,000 gene therapies for different, diseases, and hope this price tag will come down after some time. And then, of course, we are playing with biodiversity. We are playing with crops. So obviously, they have some unintended health risk if we keep on consuming genetically modified organism or plant or any fruits or vegetables, right? So those kind of bad things are also, you cannot ignore. But the ugliest thing came after this conference. The ugliest thing so far in genome engineering. So this conference was taking place in November 2018. And then He Jiankui, a scientist, Chinese scientist, he came and gave a talk, right? So this actually kind of changed the whole world, genome engineering world, I would say. So what Dr. He Jiankui explained, that he basically for the first time, basically gave birth of 2 genetically modified human babies. And they named like Lulu and Nana. So this is the chronicle of Lulu and Nana. So what that scientist he did, he kind of modified CCR5 gene using CRISPR-Cas9 system in the human embryos, and they kind of gave birth to 2 twins, Lulu and Nana. They were born, and what his hypothesis was that once this particular gene was mutated those two individuals would never get infected with HIV virus. Okay, fine, good. But that was, first of all, not a published data, just he mentioned in the conference. And second thing, nobody knows what actual experiment was done, what was the actual goal. And that kind of brought in a lot of controversies across the world. Is it really essential to change that CCR5 gene to inhibit HIV infection? No one knows. And no one knows where Lulu and Nana are now, as of now. So we do not really know what's happening. So basically, this kind of changed the world, and we can ask that, okay, if this is allowed, are we actually ready to adopt or ready to, bring in designer babies? So if this is allowed all over the labs, tomorrow, at IIT Guwahati, somebody will be sitting and designing a baby for someone, right? Let's say, appearance like this, unique personality like this, muscles like an athlete, or, swimmers, muscles, something like that. So are we actually ready for those kind of things? So this is the biggest challenge nowadays. And this brings in the most important debate, whether we should stick to somatic cell genome editing, or we are allowed to go for germline genome editing. So if you are doing a germline genome editing, that means we are modifying genetic material in the oocyte or the sperm. So basically, we'll have a modified embryo and a modified human being, right? And that is done in the genetic material of a sperm or ova. So basically, that will be passed on to future generation. So this brings the most important question: are we ready, or should we allow that kind of modification? That is the biggest challenge. We do not appear, of course, so far that is not allowed. And I found this talk yesterday. It was kind of an interview with Jennifer Doudna, who got the Nobel Prize for discovering CRISPR gene editing technology. It is just on 28 August that interview was, and she says that, okay, we should have a limit of editing human embryos. We cannot stop it. Probably in future it's going to happen, but there must be a rule or regulation for this kind of technology. So this is ongoing debate. Probably FDA or those government organization, they are going to, worldwide, they are going to get some, specific rules every other day changing. The last thing I'm going to tell today, the CRISPRing in your garage. This is something totally new, and I found very recently. So this is kind of a DIY biohacker, and this is kind of bringing in new challenges. So do it yourself. Nowadays, everybody is using, social media, Instagram or Facebook, upload their own video, they dance, they sing, they play something. So it's kind of a DIY is going on. So what happens? This person, Zoshia Zainer, so he's in US. So he himself claimed to be a biohacker. So what he is doing in his garage, he has all the materials to modify a genome. Imagine what is he doing now. So he's holding a biped. All these things are happening inside his room or in his garage. And what he did, he injected CRISPR-Cas9 entire tool system into his skin. And his hypothesis was that, okay, he could change the skin color or not. And trust me, many of those companies can sell or selling those kind of toolkits, which is very, very cheap, online. And they are kind of selling those things. This guy, he himself has a company. So if you have this kind of tools, anybody can have that tool, and anybody can have a lab of their own in their home or anywhere basically. So this is kind of a new challenge, and laws should be passed, legal action should be taken, or a proper guideline should be there, what to do with this kind of technology or this kind of hypothesis. Okay, so in summary, I'm not going to give you a list of bulleted summary, but overall, genome engineering, it's been done for ages. It's been done. It was slow, but over the last 7-8 years, it's kind of exponentially growing. A lot of crops and different human diseases are being treated nowadays, and we are successful to some extent. And we are going to have better tools day by day. Almost like 10 different Cas9s are being developed, especially in certain institutes. So those are coming in. It's now available everywhere. But all we need a kind of cohesive cohesive approach to control this. Otherwise, one small editor for human can, change the entire humankind. So proper technical controls and controls, like if there is an endonuclease which always acting, we have to stop, we have to inhibit that kind of endonuclease. Probably we'll be injecting some antibody against Cas9 to inhibit at certain point. So basically reversibility should be there. Then there should be surveillance. And of course, how to control the containment. And of course, end of the day, governance. And it is not a laboratory or an institute or a country based. It should be an international governance. How to, use this kind of powerful technologies that could actually change the entire human civilization. And trust me, it is going to happen if there is no, clear and strict rules. So with this, I will stop. I think time is already 10:30. Thank you very much once again to all the attendants. Hopefully, you can think about it. This is not something very complicated presentation, just kind of a thoughtful things. So you yourself can think what should be done, what should not be done when you design your next CRISPR experiment. And thank you once again, Professor Bora. And if you have any questions, let me know.
Yeah, we can have some questions. I can see some questions popping up. Am I audible?
Yeah.
Yeah. So can you see the chat box over there? Or shall I read it? So one question from Sam Elrin. He was asking, any off-target issues concerning plants, sir?
Sam, it's not on my fingertip, but I am sure when they're playing with different crops and unwanted specificity, there are certain examples, I'm sure. And actually, I was reading this paper on crop, drought-resistant crop, and also with the cattles with tuberculosis resistant, where they knocked down NRAMP1. There were some off-target effect. They saw some issues with some of those animals, in cattle especially. There are, but with crops also I see there are certain changes. So I need to really look into it, but I'm sure there would be, because technology is more the same. It's not something totally different. Yeah.
Yeah, next question is by Dr. Geeta Bharati. There are over 6,000 single gene disorders, the vast majority of which have no targeted clinical treatment. Is it ethical to test for such variants?
Yes, Dr.
No,
Geeta.
she's telling it is ethical to test for such variants. I think it is not a question, it is a suggestion.
Yeah, it's a suggestion. That's a good suggestion. And I began my talk with that. Actually, that was the goal, and that should be the goal. We need to. But thing is, it's again somatic versus germline. If you are treating me tomorrow, if you're injecting me, I have a genetic disorder with all the cocktail injecting me, and if I am cured, well and good, welcome, do it. But it should not be propagated to the generations. What Lulu and Nana, right? They were not signed. They didn't sign that consent form when that scientist, their parents probably signed that, but they didn't sign that. So what next generation from Lulu and Nana, they are going to have the same mutation or not? Lulu and Nana they did not get the chance to, sign that, and they didn't know. So those kind of issues should be, restricted. But otherwise, yes, rare disease, we need this.
So there is another opinion from Sonali Kelkar. She's telling it should be allowed only for hereditary diseases. Probably she's telling about the therapy part.
Yeah.
Yes, and she has some words of appreciation.
Oh, thank you.
Wonderful session, sir. Others, please kindly type some questions. In the meantime, I will read the opinions of other participants. So Dr. Shinosh is thanking you very much for the interesting and informative session.
Thank you.
Dr. Ajay Kumar does also have some words of appreciation for you.
Thanks.
And he's thanking you for the informative lecture.
Oh, thanks. I'm
and
glad.
Also Sam Elrin is thanking you. Dr. Geeta Bharati is writing highly informative lecture. Thank you, sir.
Oh, thanks.
Dr. Jagadish is writing Really great session. Thanks. Yeah, there is one question, it seems, from Raj Beg. Genetically modified plants cultivation has not picked up due to some side effects. Is it true?
See the slide that pie diagram I showed. In countries like US and Brazil, 40-50% are now GMO, but in India it's still little less. Probably person from that field can tell what actually happening, why it is less. But I think it's going to get up. It's not that always going to be like less. People are trying, I think. But remember, it's not always good to have any GMOs, right? Nowadays, people are searching for organics. So it should be a balance. It should be a balance. In countries like here in US, you really need to see what it is, what plasmid you are eating. We don't really know. So it's we need GMOs, crop-based GMOs, but to certain limit probably, yes.
Yeah, Dr. Jagadish is writing really great session, sir. And also Rajiv Kumar Singh, Asa Pai, DC Bhattacharya, Priyadarshini. They are sending you the thank you here. One question is coming up from Sogata Saha. Among different editing platforms, which one has the lowest off-target effect?
As much as I know the literature, actually talents are probably better than CRISPRs in terms of off-target effect. But again, nowadays CRISPR-3, CRISPR, in case instead of Cas9, you have SP-Cas9, you have Cas13 or 12K and all those things. They are being developed, so I think it will be something in balance. Yeah, but so far, I mean, we use both talents and CRISPR-Cas9, and I think same issues. There are certain off-target, sometimes not, sometimes not. So only thing, I think what I would suggest, Dr. Saha, is the thing is you do this technology, but post-editing screening is really important. Usually what we used to do, we send the cells, some of those edited cells for karyotyping. The karyotyping is kind of, that was done in like 1,000 years ago. It's not something very efficient. So nowadays we'll be sending it for whole genome sequencing. So that will tell us, what's actually going on. Bioinformatic tools are being used, but that is again not optimum.
Yeah, Dr. Deepa Bhamani is telling you thank you. One question from Dr. Neeraj Singh. In case of multiple gene regulating character, how much this CRISPR-Cas9 system is efficient?
Thanks, Dr. Singh. Great question. But honestly, I do not have an answer for that. If you have multiple genes, obviously you cannot target all the genes at the same time. So those type of gene editing has not been tested as far as I know. One gene like whatever SMA and all those muscular dystrophy, if it is a chunk of DNA you are removing, that is fine. But if your target gene is one chromosome and another is another chromosome, then I do not know. It's a mostly hypothetical scenario. I do not know the literature, but I would believe if you are injecting Cas9, how much Cas9 you will be injecting? Let's say your guides will be different. That's perfect. But again, it depends on how much Cas9. And sometimes, let's say in that healthy six-pack rabbit story, they injected two different guides for one mutations. Now, if you are correcting, let's say, two genomic disorders at the same time, you will be injecting like 10 different guides. So I'm not very sure if that will be efficient, or it will be efficient if you correct one and then correct the second one. So yeah, I do not have a clear answer for it.
Yeah, I think that is the next frontier to conquer. I mean, not immediately.
Yeah.
I think, after 50 years or maybe 40 years, maybe within 20 years, a lot of research can be directed in that direction. I think there is a similar question from Pinku Moni Talukdar. She is asking, what about polygenic disorders? I think it is a similar question.
Same question.
Yeah, will we be able to use editing in such cases?
Yes, we can, but again, we need time and more in vitro assays with those kind of cells. And I think the combination of gene editing and that stem cell biology, it's kind of opening up a lot of avenues. It's just the beginning. Wait for 4 or 5 years, you will have a lot of answers.
There are some other questions. Komal Taneja is asking, considering the development of this technology, there must be some successful human genome editing cases also.
Yeah.
Could you please tell about some papers? Oh, I think
mentioned
you
that
just discussed about that Chinese case. Yeah.
Yes.
Please go ahead. Yeah.
Yeah. So no, I just mentioned, and some of the successful is like SMA, like that spinal muscular atrophy and muscular dystrophy. Those are successful things going on. I mean, those are good papers, and already it's in clinic. But those are again somatic cell mutations, but we are correcting, not the germline one. And for germline one, we are actually funding agencies are actually regulating it. So in US at least, or in UK, Wellcome Trust and European Union and NIH, they do not fund anything related to germline editing. They have stopped it. There is no question of, in fact, proposing it. So it has stopped. Thing is, somatic cell, they welcome, but with a lot of restrictions. But yes, papers are being published and it's in clinical trials. Yeah.
So participants, we have a lot of time. You can take this opportunity to ask Dr. Das as many questions you like. He will try to answer with the best of his capabilities. So I have a friend from my a question from my friend, Judy Dihinia. So she's asking, in US, people have been consuming GM food. Recently, GM salmon also came to market. Are those tested for off-target effect?
I do not know, but in theory, they have been tested. It is not just randomly they will transfect some gene editing tools or plasmids to have better salmon growth or have more meat in it. They have been tested. Without testing, in FDA is not going to allow anything to commercialize. But again, Juriba, the thing is, the paper I just showed you from Kat Nathan from Crick Institute in UK, they did the experiment in 2017. They were happy, no unintended side effect, everything safe. They pinpointed what mutation was done. But after 3 years, they used a different technology just to detect what they did, and they found heterozygosity was lost. So the salmon, what we are consuming today, may not have any side effect or off-target effect. But after 5 years, you just enter your salmon meat in a genome sequencing machine, you will have certain deletion or insertion or some recombination. We do not know, but I believe whatever FDA proves, or Food and Agriculture Department, whatever it is there in US FDA, I think they'll definitely take care of it.
There's another question from Jayanti. Yeah, you need some break or some water, Dr. Das. Help yourself out there.
No, it's fine.
Yeah.
Totally fine.
Yeah, nice. So Dr. Jayanti is asking, talking about genome editing and environment, how good it is over GMO? That is question number one. How does countries saying no to GMO consider these technologies? Okay, that is the follow-up part of that question.
So how is it better than GMO to gene edit it, right? That's what the question is.
She's asking whether genome editing or engineering technologies are better over GMO, genetically modified organisms, with respect to the environment.
I would say precision thing here. So genome editing, whatever the enzymes and those things are coming, they are, I would not say they're bad, they are actually precise. So your editing technology is definitely better than what traditionally done last 20 years ago for GMO production, right? Or plasmid-based things, what was being done for recombination of plant or animals. But I believe with this new technology, safer would be to again use which you know better. It's a balance right now, whereas gene editing technology, especially CRISPR, it's rapidly evolving. So I would say, so far GMO-based edibles are better, but with time goes on, I think rest of the gene edited technology or the crops or meat or whatever you want to consume would be better. But so far, that's why so far, everywhere, like 40% of US food is GMOs, not gene edited. So, but it's going to replace it.
So there's a question from Dr. Panav Kumar Nath. I know him personally. He is a scientist in the NRL biofuel division, which they have bioethanol division, which they have started recently. So Dr. Nath is asking question regarding, sir, kindly usher some info on industrial utilization of engineered microbes for bio-based chemicals production and risks associated.
Thank you, Dr. Nath. So, like as mentioned in one of the slides, many microbes are being targeted. And for any technology, if you want to develop, microbes or prokaryotes are the best model to begin with. So you have this good probiotics available, which are GMO modified or gene edited bacteria or virus are there. But in terms of biofuel, I think people who are in this field who are actually using those kind of microbiomes can explain better. But I personally believe, let's say fungi or those things are being, or water hyacinth, those things are probably being used for, generating energy sources. I do not know which part of them will be utilized for actual fuel, generation, but if it is something related to leaves or roots or some other, organelle from that, I think this kind of modification could definitely bring in certain help.
Yes.
But so far,
I think synthetic.
I do not know. Yeah.
Please.
No, I think you can
I think
better explain.
synthetic biology and metabolic engineering are now largely benefited by the genome editing and engineering technologies. So a lot of work going on in that area. And I think the commercialization aspect, the industrial application will be equally dominating with, versus the therapeutic applications. It's a very important question.
Yeah.
There is another question from Dinesh Prasad. He's asking, sir, can we think CRISPR-Telen as future medicine?
Oh yes, Dr. Prasad. Yes, Prasad. Yes, we can, and that's what I explained. I mean, it's going to happen. It's going to happen. It's happening. CRISPR-Cas9, but again, depends on the specificity, different safety. But I think it's going to happen, and it's happening actually. Yeah. But again, the question comes to affordability. So, I mean, if one medicine, one single shot of this SMA CRISPR tool costs 2 million, right? So imagine in rupees how much it would be, right? So that is the problem. So again, let's say tomorrow I kind of produce, or let's say countries like developed countries like United States or European countries, they use this technology. They use this technology to have some better crops. So you have some unexplained problem. What will happen to people or the farmers in India or Bangladesh or Southeast Asian countries who cannot afford this kind of technology? So there will be an economic imbalance, right? That is something really need to think about. Forget about countries. Let's consider India, right? Let's imagine in a state, if government allow or some companies distribute 1,000 kits to modify one gene and have better maize or rice production, and farmers in some other state do not have that, then there will be a problem to those poor farmers. So these kind of things, whether it's a human therapy or it's a kind of a crop production, those kind of legal issues one should consider. And I'm sure government is doing that, working on it.
Dr. Das, I was thinking something just before this question pop up. So you spoke about that DIY kit, the person developing in his garage, and also he is able to do some work. Now in future, will such kind of biohackers contribute towards cutting down the cost?
True.
Or there will be some kind of, real problems. I mean, the kind of, jhola sap doctors we have in India.
Absolutely.
So is something of that kind also going to emerge?
Absolutely. That's a new challenge. It's a completely new challenge. It's not that off target. It's a really, really, dangerous situation. And he's
Right.
saying his company is called Odeon or something. He said, okay, I'll send you, just give me the money. We'll send you this particular kit. Do yourself. And he really believes. And he's, of course, he's a PhD in biophysics, but he kind of thinks that he can do it by himself, injecting GFP gene on his skin. So he is doing that. And who knows how many people are doing that?
Yeah, and actually, after genome editing, no any evidence will also remain, because
Yeah.
no any markers, nothing left over there.
Nothing.
And it will be very difficult to discriminate whether the genome edited one is actually the wild type. So these are real challenges, and I think regulatory need to be very, very strong.
Very strong.
But of course, I think creativity should also be nurtured.
Yeah.
But I think there has some kind of control. It's good that molecular biology can become a citizen science project or program. But I think definitely some kind of regulatory is required in this aspect. I move on to the next question. Dr. Ajay Kumar Das is asking, sir, what about gene therapy for some commercially important plants? Probably it's not a gene therapy in plants, or maybe, I don't know. You can interpret the question yourself. What about gene therapy for some commercially important plants?
The example I gave you, I think that knock-in or knockout of one gene from mushroom, that actually inhibited the mushroom immediately within a day or two. If you buy, it will become brown and it's just slippery and sticky. So they modified this mushroom, modified certain genes using gene editing tools, and it's kind of lengthens its life. So such kind of commercial things are going on in any other crop. So it's doable, and people are doing it.
Yeah, right. And then I think it is one of the fastest product to come to the market.
Look at the number of papers. Look at the number of papers. That's why it is coming up, right? Look at the number of papers per year in last 6-7 years.
A few years, 1 or 2 years back, I read this news. That mushroom is already in the market. It is in the shelves. Yeah, right. And it was developed very quickly. So whereas a GMO would require a long time.
Long time.
Yeah. So these are all matters of policy and regulation. There is another question from Komal Taneja. Sir, is there any example for neurological disorder with this technology?
Komal, actually, we are doing ourselves in our lab. We are working on Alzheimer's disease, and also we are working on Down syndrome to some extent using this kind of modification. But many other labs are doing. I mean, especially where you have mutations in certain genes. Let's say, for example, you have Huntington, right? So you have an extended Huntington protein that leads to problems. In Alzheimer's, even though it's a sporadic, but there are genes called APOE, alleles are there. So in human, 3 different APOEs are there, APOE2, APOE3, and APOE4. So if one individual gets 2 alleles, APOE4, APOE4, then that person is almost invariably going to get Alzheimer's. But if that person is having APOE3 isoform or 2 isoform in their allele, they are almost safe. So what people are trying to do, at least in lab, on a dish or in a mini dish or an organoid, brain organoid, we are trying to correct that APOE44 allele to APOE34 or APOE43 allele or APOE2 and 4, right? That combination, so that those cells do not show Alzheimer's like pathology. That's what we are doing. Let me give an example. So in our lab, in our university, what we do, we use the stem cell technology and gene editing technology. So we are basically extracting fibroblast from Alzheimer's patient. So once we have the Alzheimer's patient fibroblast, we convert them to iPSCs, and from those iPSCs, we generate neuron. So while generating neuron, we can induce different mutations using CRISPR-Cas9 technology or TAL, that's every other day we are doing in our lab. So our goal is to generate certain naturally occurring mutations that actually, prepone Alzheimer's pathology or exaggerate Alzheimer's pathology, just to see how we can manipulate those kind of genes. So in neurological disorders, yes, the example I gave you, basically that AAV9 we injected in mouse brain, it went and it actually eliminated amyloid beta plaque from those mouse in our own paper. Many other groups are doing Down syndrome or Huntington, but it's rapidly growing. It's any neurological disorder, yes, it's important because many drugs do not cross the blood-brain barrier. So this kind of technology, especially AAV9 isotype, can manipulate genes inside the brain. So it's important and it's ongoing.
Yeah, thank you, Dr. Das. So there is one more question. I think we'll take this last question and no further because we are just about to be 11. Our next speaker is ready, as I have come to know. We'll have that session, of course, at 11:15. We'll have a 15-minute break. Okay, Kamal Thaneja is asking another question. Any case for Fragile X?
I do not know, but I think there would be. I'm sorry, Kamal. I do not have that answer, but I think it will be there. DMD is being, doing similar things. So Fragile X will be, or it's being already targeted, but it's not on my fingertip.
Yeah. So thank you, Dr. Das. It was an excellent lecture. And I see a lot of appreciative words from the participants.
Thank you so much.